So first I want to point out that the results that I'm going to present today are from experiments I've done with Chris Cornwell, so it's not only my work. As Malcolm pointed out, we have like a very good knowledge now on how corals are going to respond to changes in pH under constant condition, but we really don't know what's happening when we start to play with pH variability. There are maybe like 2 to 4 studies done on corals for this and even less on coral and algae, so it's really something we need to work on. And pH variability can be very interesting because pH variability is caused most of the time by the organisms themselves. So with photosynthesis, they will increase pH during the day and respiration tend to decrease pH at night. pH variability can be found at very different scales. For example, at micro scale, we can have changes in pH in the layer of seawater that is directly in contact with the organism. So it's called the diffusive boundary layer, which is a layer of seawater where the molecules are transferred only by diffusion. So in this layer, any metabolic activity will tend to really change the pH condition rapidly. But pH can also vary at larger scale. For example, on reefs, those measurements represent pH measured on Talon Island, which is the reef in the Kimberley where we work. And on this reef, pH can change up to 1 unit per day, so which are like really crazy large changes, and it's maybe one of the largest change reported in the world, so this reef is really very interesting for that. So what we wanted to do with these studies is to test if pH variability at different scale affect the response of organisms to ocean acidification. So are organisms exposed to this variation in pH more or less resistant to the changes that were expected by the end of the century? We also wanted to determine if environmental history can affect the response to ocean acidification. So are organisms living in those environments more or less sensitive to ocean acidification? So to respond to those questions, we have done 2 different lab experiments. One in which we try to look at the role of the bulk seawater pH variability in order to reproduce, for example, what we saw on this reef in Talon Island. So what happened to organisms exposed to these big changes in pH? The second experiment, we also wanted to look at a different scale and try this time to create this pH variability by changing the condition in this diffusive boundary layer. So for that, we play with the flow in order to change the thickness of this layer, and we also play with the light level in order to affect the photosynthetic activity and then potentially the pH in those conditions. We also have done a lot of field work. We went in the Kimberley, so on those reefs with stable or variable condition in pH, and measure the response of organisms and reef to those different conditions. But I will not have time to talk about this today, so I'm really going to focus on the lab work. So like I said, our first experiment was to look at the effect of the bulk seawater pH variability. So for that, we went in the Kimberley. Like Verena explained yesterday, the Kimberley region is very interesting, this micro-tidal regime. And with these different tides, like some reefs are really completely separated from the surrounding seawater at low tide. So if you look on the top right photos, you can see Talon Island at low tide. So the reef really on top is several meters above the seawater. And this reef is very interesting because it's covered with seagrass, with macroalgae, and therefore all those organisms with their photosynthesis or respiration are going to change the pH condition very rapidly. So what you can see also on this graph is then the pH measured on Talon Island. So we have these very large diel changes with very high pH during the day, very low pH at night, so pH much lower than what's expected by the end of the century. And then we have another site where we work, Shell Island, where the pH conditions are very stable there because this tide pool is dominated by corals and not by seagrass and seaweed, where more an effect on pH. So we went to those sites, we collected corals and CCA, the same species that we found at the 2 sites, and brought them back in Perth in our new laboratory to work on the effect of pH on those organisms. So what we tried to do is recreate those conditions in the lab. We had like ambient conditions, so stable pH to represent the condition in Shell Island. We had ambient fluctuating condition where we wanted to represent the condition on Talon Island. And then we had some treatment where we applied an offset representing the condition expected by the end of the century. So in the graphic on the bottom that you can see here represent the pH that we obtained in our tank. So we're pretty successful at reproducing those cycles of very low pH at night, representing low tide at night, then an intermediate pH representing the mean seawater condition, and then a high pH during the day representing low tide during the day. So I'm going to jump directly to the results. So those results represent the mean classification for corals after 6 months in the lab. So we try to do like a pretty long-term experiment, and this experiment is still running, so maybe the results are going to change in the future. So on this graphic, what you're going to see on the left side, it's the result for Shell Island, so where the pH conditions are constant. On the right side, the result from Talon Island, where the pH conditions are much more variable. So what we observed that was pretty surprising, it's finally there is a very similar response for the corals coming from those two different environments. We can see the highest classification was measured in those like ambient fluctuating conditions, so they seem to really like those fluctuations. And the lowest classification rate were found when we have like a low pH but under constant condition. If you look at the orange bar, they represent the ocean acidification plus fluctuation in pH. And corals were able to maintain a relatively high classification, almost the same rate than in the ambient condition. And this is surprising because pH was down to 7.2 at night, so it's like very, very low pH condition, but they're still able to calcify. So that's very interesting. For the coral and algae, we observed something totally different, since for them, really there was like the highest classification was for the coralline coming from Talon Island. So they are used to those conditions, used to the fluctuation in pH, and we can really see that in the lab. They really like those conditions, they like those fluctuations. We also observed there was not really a very strong effect of pH on any of those coralline calcification. So for the coralline, what we did, this result represents the calcification of those adult coralline after 3 months in the lab. And we realized when we were in the lab that they were recruiting quite a lot in the tank. So we were like, okay, let's maybe now try to work on the recruitment of coral and algae and try to work on different generations rather than to focus on the adults. So what we observed, this is the recruitment of coral and algae after 3 months in the lab. Then that's like our first generation. And then we had like also some very interesting results with like this time like higher recruitment rate in Shell Island for like with those big fluctuations in pH. So that was like a bit unexpected. But what was really interesting, I think, it's the fact that recruitment rate was really low when we were in those conditions with those big fluctuations in pH, and on which we applied this offset. So those are the results for the first generation. We are now running the 5th generation, and it seems that this trend is really getting confirmed with like recruitment decreasing in those ocean acidification fluctuation treatment. So basically what we saw with those experiments, like just a little bit complex, but it seemed at least that for corals, variable conditions ameliorate the effect of ocean acidification on classification. There was also no environmental history, so it was like maybe a little bit surprising, but it seems that corals coming from 2 different sites, really the same response, they like those fluctuations, so maybe to have high pH during the day, it's enough for them to compensate for the low pH at night. For the CCA, the response was very different, so this time there was really more this site-specific response that we didn't find on the corals, so maybe that would represent an effect of environmental history and the fact that coralline can respond much more quickly and adapt more quickly to their environment. So like I mentioned, the experiment is still running. We're at the 5th generation of recruitment for the coralline. We're going to keep doing that maybe for another 5 generations in order to really try to see if coralline can maybe adapt a little bit or change their classification of that in those conditions. We're also working on determining the pH in a calcifying fluid, GIC in a calcifying fluid, so Malcolm introduced that before. And the goal would be really to see, okay, how can we link the pH variability in seawater with what we measure then in the calcifying fluid? And can we, with this kind of measurement, explain why corals under those big fluctuations in pH are less sensitive to changes in mean pH? Okay, so that's for our first experiment. So the second one, we wanted really this time to just try to let the organisms do the job for us and modify the pH condition in this diffusive boundary layer. So what we did, we had the chance to have these new facilities, and so we were able to build those 12 flume systems. So they're about 2 meters long, and we can change the flow condition. We have a good like pH control, temperature control, so it's a pretty nice setup. And the goal was really by playing with the flow level, which we were able to change the thickness of this diffusive boundary layer. And then adding on top of that the effect of light to change the photosynthesis, we are really trying to modify the pH in this layer. So what we did, we worked with 2 different flow speeds. This time we maintained the pH condition constant. We had 2 different pH, 2 light level, and we work on corals and coral algae that were coming from Rottnest Island, just offshore of Perth. So for the results, if we look, this is for the coral acropora. What you will see on those graphics, it's the 4 bar on the left represent the high light condition, and we have low light condition. Then the 2 different flow level, and then this is crossed with different pH, so sorry, it's a bit complex. So what we observed is that light was finally the main driver for those corals, with like much lower calcification under low light compared to high light, which is not really surprising since they come from a relatively high light environment. What was interesting is that we observed like a combined effect of flow with CO2. So when we had like the lower flow condition, we had a stronger effect of pH on those organisms. So this was maybe a little bit unexpected. What we saw also, it's under the high flow condition this time, there was really like no effect of pH on the calcification. So then we work on another coral, Plesiostrea, which has a very different morphology, so we wanted to compare branching coral versus massive corals. And on this coral, we observed this time there was like no effect of light, but there was this time an effect of flow, with calcification in general being lower when we decrease the flow. These corals were just like not sensitive at all to ocean acidification, so that would go in what Malcolm was talking about. Maybe ocean acidification is not that bad for corals, I mean, at least for this one. For coral and algae, this time it's a very different story, and for them it seemed that pH condition had a very strong effect on them, where like calcification was close to 0 under the low pH conditions. We also observed that there was an effect of light, strong effect of light, and really like when we had like low light condition, low flow, it was really hard for those coral and algae to be able to catch up and calcify. They even tended to dissolve in the lab. So what we show with this, it's like it really seems there is like for coral a species-specific response to flow, and this is probably linked to the morphology of those corals, difference between branching, between massive corals. So it's something very complex, not as easy as we change a little bit the condition in the GBL, and that's going to help or not the corals. For the coral and algae, it's also like it seems there is this strong effect of flow, so low flow, low light, there's really a lot of problem to catch up with this. So what we're going to do now in the next week is to really try to determine this pH in the diffusive boundary layer, so on this very micro layer. So we're using pH microsensors. It's pretty tricky because we have to get like really close to the surface of the organism, and those sensors are really easy to break and very expensive, but we are working on that, so that's going to be really interesting. What we're also trying to do is to look at the link that there is between flow and all the physiological parameters, so calcification, photosynthesis, respiration. So we are going to do a lot of incubation under different flow to really try to understand why we observe those results. And finally, the ultimate goal will be to, like, using boron isotopes to determine the pH in the calcifying fluid, to try to link what is important for the pH in the calcifying fluid. Is it the mean pH in seawater, or is it the pH in this diffusive boundary layer? So really try to understand a bit more how all those different parameters like work together. So to conclude on this study, what we found is that there is really like a complex interaction between the pH in the bulk seawater, pH in GBL, the measured response. So as usual, it's not as easy as we thought at first. We thought it would be pretty straightforward, but it's a very, very complex system. In general, it looks that there is a positive effect of seawater pH for corals. This has been shown before in like the only other few studies that have shown that. So it seems that pH variability could really help the corals to deal with ocean acidification. We didn't really see any strong effect of flow in our experiment, so maybe we're not like our flow regime were too close to each other to really measure something. This experiment is also still ongoing, so hopefully in the next months we'll maybe be able to like tear apart those different effects with like a bit more time for the organisms to respond to that. So we are, like I say, we also did a lot of measurement in the field, so we are trying to link those experiments that we do in the lab with what we measure in the field. So we go on those reefs where pH change very quickly, and we can measure the response of organisms to those conditions, but also to the reef, so at the reef scale, or it's the entire reef responding to those conditions. The first results are very interesting. We have really these big cycles of calcification with the reef doing like really high calcification during the day, very low calcification, or even dissolution at night. So even if it seems that pH variability doesn't really affect mean calcification of corals, when then we look at reef scale, it seems that it has a much stronger effect with potentially a lot of dissolution going on when we have this condition with very low pH. So that's something we are working on. And with that, I'd like to thank Verena and Marine for their help in the field and the lab, and all our army of interns who have been very helpful. Thank you.